Good morning. My name is Lord William B. Pakureb, and I would like to introduce my report about Father Gregor Mendel as a partial fulfillment for the course Genetics, as well as give you an overview of his experimental and quantitative methods. Genetics is a study of heredity, or how specific characteristics are passed down from parents to their children or grandchildren. Every plant and animal produces offspring of the same kind or type. The kids bear a strong resemblance to their parents. However, kids sometimes are not identical to their parents. Their hair color or height, for example, may differ. Genetics explains why some children inherit parts of their parents' characteristics but not others. The fact that we are studying heredity, or how strong my resemblance to your mom, is basically the fault of an unsuspecting Austrian friar playing with his pea plants in his monastery. Before we proceed with the lesson proper, for the sake of technicality, let us first define vocabularies pertinent to what we are going to discuss in this very moment. First and foremost, genetics is a field of biology that studies genes, genetic diversity, and heredity in living things. Genetics is a study of how genes and how traits are passed down from one generation to the next. Our genes carry information that affects our health, our appearance, and even our personality. Secondly, a phenotypic trait, also known as a character state, is a unique version of an organism's phenotypic feature that can be inherited or dictated by the environment. Most often occurs as a mix of the two. Traits are our physical characteristics, like our hair or eye color. Every living thing has traits that make it unique. Next, the passing on of qualities from parents to their children is known as heredity, also known as inheritance or biological inheritance. The offspring cells or organisms acquire the genetic information of their parents through asexual or sexual reproduction. Simply put, the passing down of traits from one parent to child is called heredity. Then, dominance refers to the occurrence of one gene variant or allele on one copy of a chromosome concealing or overcoming the influence of another gene variant on the other copy of the chromosome. It could either be dominant or recessive. Dominant refers to the relationship between two versions of a gene. Individuals receive two versions of each gene, known as alleles, from each parent. If the alleles of a gene are different, one allele will be expressed. It is a dominant gene. The effect of the other allele, called recessive, is masked. Finally, a gene is a basic unit of heredity in biology, consisting of a sequence of nucleotides in DNA that encodes the synthesis of a gene product, such as RNA or protein. Genes carry the information that determines your traits, which are features or characteristics that are passed on to you or inherited from your parents. Each cell in the human body contains about 25,000 to 35,000 genes. Who is Gregor Mendel? Gregor Johann Mendel was an Augustinian monk and abbot of St. Thomas Abbey in Brunn, Margravate of Moravia. Father Mendel, a brilliant scientist that he was also a faithful son of the church. Being in the order of St. Augustine, he entered a contemplative life. He rose to prominence as to become the abbot of his monastery. An abbot is an ecclesiastical title given to the head of a monastery. He was a meteorologist, mathematician, biologist, and Augustinian friar. Mendel was born into a German-speaking family in the Silesian province of the Austrian Empire, today as Czech Republic, and is widely regarded as the father of modern genetics. He was a total package. He studied the weather, numbers, life itself, while ardently living out his vows as a consecrated religious. Father Mendel lived in what was known as the Austrian Empire. After the Munich Agreement, Nazi Germany systematically took control over the Czech lands and later became an Eastern Bloc communist state following a coup d'etat. During the Velvet Revolution, the communist rule ended and Czechoslovakia was peacefully dissolved, with its constituent states becoming the independent states of the Czech Republic and Slovakia. Despite the turmoil in his country, Father Mendel, a peaceful monk as he was, out of sheer curiosity and interest played with pea plants. Who would know that this experimentation would be groundbreaking in the field of biology, as to have him to be called the father of genetics because of an unsuspecting monk crossbreeding pea plants We have now to add one subject in our course. Though farmers had known for millennia that crossbreeding of animals and plants may favor some desirable qualities, Mendel's pea plant experiments, which took place between 1856 and 1863, established many of the principles of heredity that are now known as the laws of Mendelian inheritance. Mendel worked with 7 characteristics of pea plants: plant height, pod shape and color, seed shape and color, and flower position and color. Mendel demonstrated, for example, that when a true breeding yellow pea and a true breeding green pea were crossed, the progeny always produced yellow seeds. However, the green peas emerge in the following generation at a ratio of 1 green to 3 yellow. We will discuss the 7 characteristics of pea plants in the next slide. A pea could either be round or wrinkled in shape, green or yellow in color, constricted or inflated in shape, and green or yellow in pod color. Its flower could either be purple or white, and its size either tall or dwarf. The position of its flower could be axial or terminal. Mendel used the phrases recessive and dominant in regard to particular features to explain this phenomena. The green characteristic, which appears to have gone in the first filial generation, is recessive, while the yellow is dominant in the prior case. He published his findings in 1866, establishing the role of an unseen factors, now known as genes, in defining the features of an organism in a predictable manner. We now go to the experimental and quantitative approach of Gregor Mendel in conducting his famous study of peas. Mendel grew up on a small farm in what is today the Czech Republic. In 1843, Mendel entered an Augustinian monastery. From 1851 to 1853, Mendel studied at the University of Vienna, where he was inspired by a physicist who supported experimentation and the application of mathematics to science, as well as a botanist who piqued Mendel's interest in the reason of plant variation. In Mendel's test, these factors collided. Experimentation provides us with knowledge of the physical world, and it is experiment that provides the evidence that grounds this knowledge. It is through experimentation that Mendel laid the foundations of genetics. Experiment plays many roles in science. One of its most important roles is to test theories and to provide the basis for scientific knowledge. On the other hand, mathematics is an intrinsic component of science, part of its fabric, its universal language, and indispensable source of intellectual tools. Reciprocally, science inspires and stimulates mathematics, posing new questions, engendering new way of thinking, and ultimately conditioning the value system of mathematics. After university, Mendel taught school and lived in the local monastery, where the monk had a long tradition of interest in the breeding of plants including peas. Around 1857, Mendel began breeding garden peas to study inheritance. Plant breeding began with a sedentary agriculture, namely domestication of the earliest agricultural plants, which is thought to have occurred between 9,000 and 11,000 years ago. Initially, early human farmers chose food plants with favorable features and utilized them as a seed supply for succeeding generations, resulting in a gradual accumulation of traits. However, trials with purposeful hybridization began to emerge throughout time, and Gregor Mendel's work considerably aided the science and knowledge of this subject. Mendel's discoveries eventually led to the birth of genetics as a new science. Many fallacies concerning heredity existed prior to Mendel's discovery. Two of the most common blunders were particularly deceptive. The first was that one parent is responsible for the majority of an offspring's inherited characteristics. One of the early microscopists, Nicholas Hartsocker, claimed in 1694 that it was the male via a fully developed homunculus inside the sperm. Another fallacy was the concept of blended inheritance, which claimed that parental qualities were mingled and permanently modified in children, similar to how blue and yellow pigment on a painter's palette merged to become green. The hypothesis of blending may have evolved from parents' natural desire to see a mix of their own characteristics in their children. While blending may explain offspring who seem like a mix of their children, it couldn't account for noticeable distinctions between biological brothers and sisters, or the persistence of variety within extended families. The test, designed by Mendel, would dispel these beliefs by offering exact, verifiable answers to the 3 questions presented by Ab about NAP over 15 years before. What is inherited? How is it passed down? What part does chance play in heredity? Mendel's discovery was largely due to the manner NAP set up his research. What did Mendel do differently from those who preceded him? Pea plants have several advantages for genetic study. Pea plants are available in many varieties that have distinct heritable features or characters with different variant traits. Peas have a strong generation time. Each mating produces many offsprings. Mendel was able to strictly control the matings of his pea plants. Each pea plant has male and female sexual organs. In nature, pea plants typically self-fertilize, fertilizing ova with a sperm nuclei from their own pollen. Mendel could also use pollen from another plant for cross-pollination. Mendel began by selecting the garden pea or the Pisum sativum as his test organism. Peas thrive in brun, and because they had both male and female parts in the same bloom, they usually self-fertilize. Both the egg and the pollen come from the same plant in self-fertilization or selfing. The structure of pea blossoms, on the other hand, makes it simple to avoid self-fertilization and instead cross-fertilize two individuals by brushing pollen from one plant to a female organ of another plant. Peas provided another benefit. Each generation after that, Mendel was able to produce a huge number of individuals in a short growth season. In contrast, if he had worked with sheep, each mating would have and a generation gap would have been many years. Mendel tracked only those characters that varied in an either-or manner rather than a more or less manner. For example, he worked with flowers that were either purple or white. He avoided traits such as seed weight, which varied on a continuum. Mendel investigated the inheritance of distinct alternate forms of certain features, such as purple versus white flowers and yellow versus green peas. Because there were no intermediate forms, he could discern and trace the transmission of one or the other observable attribute using such either-or qualities. Continuous features such as height and skin color in humans are the polar opposite of discrete qualities. Continuous traits have several intermediate versions. Mendel started his experiments with varieties that were true breeding. When true breeding, plants self-pollinate, all their offsprings have the same traits as their parents. Mendel gathered and propagated true breeding pea lines. Offspring from such pure breeding lines carry distinct parental features that are passed down from generation to generation. For up to 8 generations, Mendel studied his pure breeding lines. Plants with white flowers always produce white flowers in their progeny, whereas plants with purple blooms always produce purple flowers in their offspring. Mendel used the term antagonistic pairings to describe alternative features that are always present but mutually incompatible, such as purple against white flowers or yellow versus green seeds. And he chose 7 such pairs to examine. In his experiments, he not only perpetuated pure breeding stocks for each member of a pair, but he also cross-fertilized pairs of plants to produce hybrids, offsprings of genetically dissimilar parents for each pair of antagonistic traits. It's worth noting that each of the hybrids for Mendel's 7 antagonistic features resembles just one of the parents. The dominant characteristic is a parental trait that shows up in the hybrid. In a typical breeding experiment, Mendel would cross-pollinate or hybridize 2 contrasting true breeding pea varieties. The true breeding parents are the parental generation and their hybrid offspring are the first filial generation. Mendel would then allow the first filial hybrids to self-pollinate to produce a second filial generation. Being an expert plant breeder, Mendel carefully controlled his matings, going to great lengths to ensure that the progeny he observed really resulted from the specific fertilizations he intended. Thus, he painstakingly prevented the intrusion of any foreign pollen and assured self or cross-pollination as the experiment demanded. Not only did this allow him to carry out controlled breeding or selected traits, he could also make reciprocal crosses. In such crosses, he reversed the traits of the male and female parents, thus controlling whether a particular trait was transmitted via the egg cell within the ovule or via a sperm cell within the pollen. He could, for example, fertilize the eggs of a white flower with pollen from a purple blossom and the eggs of a purple flower with pollen from a white bloom. Mendel established that the 2 parents contribute equally to the inheritance since the children of these reciprocal crossings were comparable. It makes no difference to the hybrid's shape which of the parental kinds it makes no difference to the hybrid's shape which of the parental kinds was a seed or pollen plant, he wrote. Lastly, it was mainly Mendel's quantitative analysis of second filial plants that revealed two fundamental principles of heredity, the law of segregation and the law of independent assortment. Mendel worked with a huge number of plants, counting all progeny, numerically analyzing his data, and comparing his findings to predictions based on his models. He was the first to investigate heredity in this way, and his expertise in physics and mathematics undoubtedly influenced his quantitative approach. Mendel's meticulous numerical study showed transmission patterns that mirrored basic heredity rules. Mendel was an outstanding practical experimenter. He made sure that the small plants were out of the shade of the tall ones while comparing tall and short plants. For example, so that their development would not be hindered, he eventually decided to concentrate on characteristics of the pea seeds themselves, such as color and form, rather than characteristics of the plants that sprang from the seeds. He could examine many more individuals from the monastery garden's small size, and he could evaluate the outcomes of a cross in a single growing region in this method. In other words, Mendel set up a simple black and white experimental setup on purpose and then discovered out how it worked. He didn't dig at the plethora of factors that influence a prize ram's growth or the origins of species distinctions. Rather, he examined specific qualities in two mutually incompatible forms and posed questions that might be addressed by observation and calculation. Here are the references that made me go along the way with this endeavor. Now that nothing is left to explain, this would be the perfect opportunity to end this report. Thank you for listening and have a blessed day.